dear students, so let's start the today's topic, and the name of the topic is tRNA, the adaptor molecule. So the term adaptor molecule has given by the scientist Francis Crick. Francis Crick ने tRNA को adaptor molecule भी कहा था. And why it is known as the adaptor molecule? Because the tRNA working like a vehicle, it works like a vehicle which pick up the amino acids in the cytoplasm, है ना. तो tRNA का फंक्शन क्या होता है, क्यों इसको एडाप्टर मॉलिक्यूल कहते हैं? क्योंकि जो tRNA के मॉलिक्यूल्स होते हैं, जो tRNA है वो एक्ट्स लाइक अ व्हीकल की तरह एक्ट करता है और साइटोप्लाज्म में जो अमाइनो एसिड स्कैटर्ड होते हैं उनको पिकअप करने का काम करता है. As well as it is also read the specific genetic codes present on the mRNA molecule. तो ये specific genetic codes को read करने का काम करता है mRNA molecule में, and that is why it is called the adapter molecule. It is also known as transfer RNA and soluble RNA, sRNA. So it is also called tRNA as well as sRNA. So students, let's study the structure of tRNA. The clover leaf configuration of tRNA has 5 main structural regions. The first is called the acceptor arm, second is known as anticodon arm, third is called thymine pseudouracil arm, or it is also known as T-loop. Next is dihydrouridine arm, that is D-loop, and last is extra arm. So these are the main 5 structural region of tRNA or transfer RNA. The extractor arm is concerned with binding and hiding of specific amino acid, and that is why it is also called the amino acid binding site. It is also formed by the base pairing between the nucleotide of 5' to 3' end. So students, the second arm or second structure of tRNA is anticodon arm or anticodon region, which is mainly divided into 2 parts, the anticodon stem and terminal anticodon loop. This anticodon loop is made up of from 7 nucleotides with the terminal nucleotide triplets. The third structure or the third region of tRNA is thymine pseudouracil arm, or it is called T-loop. It has a basal stem and thymine pseudouracil loop of 7 nucleotides with the obligatory presence of pseudouridine. The next arm is dihydrouridine arm, that is also known as D-loop, which is also has the basal stem and DHU, terminal DHU loop, and the loop is composed of 8 to 12 nucleotides. The last arm is known as extra arm. It is also known as mini loop or little loop, and it is not compulsory that in every tRNA structure this extra arm is present. So the shape and the number of nucleotide varies in every tRNA structure. एक्स्ट्रा आर्म के द शेप और जो नंबर ऑफ न्यूक्लियोटाइड्स हैं वो वेरी करते हैं हर स्पेसिफिक स्ट्रक्चर में, एंड देयर फंक्शन इज आल्सो अननोन. इट इज आल्सो नोन एज variable loop. Let's study some additional information of tRNA molecule. The total amount of tRNA is nearly 10 to 20% of total cellular RNA. मतलब total RNA का 10 to 20% tRNA होता है. Second, all tRNA are identical in their 3' and has a CCA-OH terminus. मतलब 3' में CCA-OH terminus ही present होगा, and at the 5' end it has a G or C terminus, और 5' end पे G और C molecule, G और C terminus present होगा. tRNA are all single structure with 70 to 100 nucleotide residues. Charged tRNA serve as the source of amino acid for synthesis of peptide chain. तो जो चार्ज्ड tRNA मॉलिक्यूल होते हैं वो क्या करते हैं, पेप्टाइड की सिंथेसिस में हेल्प करते हैं, अमाइनो एसिड्स के को साथ सर्व करते हैं और पेप्टाइड की सिंथेसिस में, मतलब पॉलीपेप्टाइड की जो चेन बनती है उसको बनाने में हेल्प करते हैं tRNA के मॉलिक्यूल्स. द क्लोवर लीफ मॉडल ऑफ tRNA हैज सर्टेन स्पेसिफिक फीचर्स. The first point is there is the presence of some unusual bases in the region where hydrogen bond is absent. So जहाँ पर भी हाइड्रोजन बॉन्डिंग प्रेजेंट नहीं होगी वहाँ पर क्या हो रहा है, कुछ अनयूजुअल बेसिस देखने को मिल रहे हैं. द 3' एंड 5' एंड आर क्लोज टू ईच अदर. जो 3' एंड है और 5' एंड है वो एक दूसरे के क्लोज हैं बिकॉज ऑफ द फोल्डिंग. ड्यू टू द फोल्डिंग 3' एंड 5' एंड आर close to each other. The overall distance between the CCA terminus and anticodon terminus is same and constant in all specific tRNA. तो हर tRNA में CCA terminus और जो anticodon terminus है उसके बीच में जो distance है वो कैसा है, constant है और same है. The last point is the possible difference is distance due to the difference in number of nucleotide would be corrected by adjusting the size of extra loop और mini loop. तो extra loop या mini loop के size को adjust करके जो difference है उसको करेक्ट कर सकते हैं, डिस्टेंस को करेक्ट कर सकते हैं. सो दैट्स इट फॉर टुडे. आई होप यू अंडरस्टूड द टॉपिक एंड इफ यू लाइक दिस वीडियो प्लीज सब्सक्राइब टू द चैनल, लाइक एंड शेयर. थैंक यू.